Hi everyone, my name is Aaliyah and my project was focused on how the chemical richness of scent marks varies among different species of lizards across the globe, given their environmental differences. So basically, to begin, chemical richness refers to the level of composition of organic compounds in a species' secretions. My hypothesis was that because of the differences in the environment, and more specifically the temperature, in which the Celloporus and Lacertidae families reside, they will demonstrate differences in the chemical richness of the scent marks between each other. My results showed that the mean annual temperature of the surrounding environment does significantly affect the chemical richness of lizards across the globe. I conducted an ANOVA two-way test with replication to test the hypothesis. The test yielded a p-value of less than 0.002, which shows that there is a very significant correlation between mean annual temperature and the chemical richness of the respective lizard species. So on the next slide, I'm showing that there is a significant difference in the chemical richness of lizard scent marks. I created a graph that compared the lizard species and then the chemical richness and showed how it varies across the two different species. And I sampled 12 species of lizards from two families, the Celloporus and the Lacertidae lizard families. The Lacertidae lizards had significantly higher chemical richness than the Celloporus lizards. I conducted a t-test to see if there was any significant difference between the average chemical richness of each lizard family, and I ended up with a p-value of less than 0.005, which suggests that there is a significant statistical difference between the chemical richness of the two populations of lizard families. In this next slide, I created a scatter plot that compared mean annual temperature and chemical richness and showed how they correlated, and I found the annual mean temperature does have a significant effect on chemical richness. So I used the variable Bio1, which is one of the 19 bioclimatic variables derived by World Climate Organization, and that was what I used to conduct my research and find the annual temperatures for each environment that the lizards live in. I then conducted an ANOVA two-factor test with replication to test how chemical richness among the two species compared with the annual mean temperature of the environment of the lizards, and then my results showed that there is a very significant correlation between annual mean temperature and the chemical richness of the scent marks of lizards. So what does this mean for the context of my project? So this means that the temperature of the environments in which the lizards live in does have a very significant effect on the chemical richness of their scent marks. The abundance of organic compounds in lizard scent marks may influence how effectively they are able to communicate with one another. It is known that the abundance of organic compounds in a lizard scent marks may influence how effectively they are able to communicate with one another. When adding temperature of the given environment as a variable, it's important to know how lizard scent marks are affected. It could be the reason for variance in chemical signals between lizards in different parts of the world given their environments. So this next slide shows the data from the data set that I used. This is from the VERSE dataset, which was titled Volatile Fatty Acid and Aldehyde Abundances Evolve with Behavior and Habitat at Temperature in Celloporus Lizards. So the data I used from that first dataset was the 12 species of the lizards that were provided in the paper. Each of the species of lizards had a sample size of 6, and all these lizards come from the Celloporus family of lizards. I was focusing on the chemical richness of the scent marks of each species of lizards. The chemical richness of each species was provided in the dataset in a column labeled richness, and because the average of each sample wasn't given, and I was given the richness of each sample size instead, I took the average of the chemical richness of the samples provided for each species and used that as my value for chemical richness for the respective species. And the mean annual temperature was also provided in the data set for each species sample, and I averaged the given temperatures for each species sample to provide a value for that as well. So I took a standard deviation of each so that I was able to do that. And for my second data set, it was titled How Environmental Conditions Shape the Chemical Signal Design of Lizards. So for this data set, I will be focusing on 12 of the lizards. In the dataset, I used a random sample because there were 64 lizards given in the dataset, so I used a random sample of 12 so that the data matched up for my ANOVA test later on. I also used the chemical richness that was provided in the data. It was labeled in a dataset in a column labeled chemical richness, The mean annual temperature for this family of lizards in this study was not provided. So to find the mean annual temperature, I calculated using the annual mean temperature formula that is provided on the World Climate Organization website. And that's all. Thank you, and I hope you enjoyed.